Okay, so our book in this first chapter, it's introducing the kinds of jobs, what biotechnology is, how it kind of impacts impacts society. So it's going to also explain the difference between university and government labs, and like manufacturing and industry. So we just talked about industry and you saw the dollar signs. Okay, so it's about making money. So university and government labs, we're nonprofit. Whoops, sorry, didn't hit the pencil. Okay, so we're nonprofit. So for the most part, right, we're not making money. We're making enough to employ the people and to keep doing research or, doing what we need to do. So all of our funding comes from competitive grant processes, or it comes from the state, the county, the federal government. Okay, so competitive grant processes. So we do grant proposals, and the grants can come from industry. Okay, so maybe they come from like MedImmune or someplace like that. They can come from foundations, maybe like the Bill Gates Foundation, someone that has a lot of money. There's all sorts of foundations that you kind of hear. The Gates Foundation is a big one, a good example. Like the Gates Foundation is always working to improve health in third world countries. And then they can come from the government. Okay, so the government, they could come from the National Science Foundation is where we've had grants from. So National Science Foundation, they could come from NASA, they could come from NIH, the National Institutes of Health. Okay, so these places take the money and they distribute it to projects that they think are worthwhile. And it's not just done willy-nilly. It's a very rigorous process. Okay, so applied science means that the science has to be able to be used. We're not just doing, especially in biotech, well, that's not necessarily true. We're not just doing science in order to do science. That rarely happens anymore. Okay, so you got to have a purpose. So we might research what causes a disease, and we're trying to understand the pathogen so we can understand how to fix it. We might look at how the host responds to the pathogen. We might be trying to develop better food sources. Whoops, I spelled forensics wrong. We might be looking at forensics. We're trying to figure out, okay, who did it? Who is the baby's dad? Blah, blah, blah. That kind of thing. Okay, so what do you need to do these jobs? So prior to 2005, most jobs in biotechnology required at least a 4-year degree, and they could have been in any of these fields. Okay, so biochemistry, molecular biology, genetics, biology, chemistry, and then biotech. Okay, but what's happened is that the industry's really grown. Okay, so it's grown from just research and development into manufacturing, sales. Okay, we have to make a lot of product. So there's lots more job opportunities. So the companies are starting to really look at people who just have a 2-year degree, and they can go in as a technician. So what they want is somebody that's been trained in all of the processes and they have some experience. So like the program at HCC, we're going to train you. You're going to take basic biology, basic chemistry, but we're also going to do more applied stuff, and we're going to make sure you have an internship experience. So you've worked in a lab, you have hands-on experience. This 2-year program sometimes is even better than the 4-year one because you get this hands-on experience. If academic degrees and titles, not everyone is familiar with this, so we'll just kind of explain like the hierarchy, how this works. Okay, so a lab assistant is someone that might have a high school diploma. They've got training. Okay, so they can go in and do what they need to do. Okay, a technician is going to be someone that probably has a 2-year degree. Okay, or they've community college. Someone that's a research associate would be someone that has a bachelor's degree. Sometimes this person is called a technician though, so that kind of goes back and forth. Okay, so after 4 years of college, you can go get your master's degree, which is probably a 2-year program. Some of them are 18 months now. And you can go in and be a research associate. So you're going to have more lab experience at this level than you would at, say, this level. Okay, so after you get your master's, you could get your doctorate. So your doctorate would be between, it depends, 4 to 6 years after your bachelor's. Some people go for straight from bachelor's to here, and they skip a master's degree. Okay, and then they can go in as a scientist. Okay, sometimes after the doctorate degree, you can't find a full-time job. So you go and you do a postdoc. Okay, and you do the postdoc, you work in someone else's lab, you work on their projects, you gain a lot more experience of like administering a project, being in charge, and then you can go get a job. Okay, you guys probably figured this out, but it's important to realize that as you get more education, you're going to make more money. Okay, normally. Okay, so there's all sorts of different types of careers in biotech. Okay, so bioengineering, so you're taking engineering and biology and putting them together. Biochemistry, manufacturing, you could work in the pharmaceutical industry, ag, forensics, chemists. Bioinformaticists, this is the big push right now. Bioinformaticists these are going to be the computer analysts, so they're going to be able to use computers, databases, algorithms, and they're going to be able to take it and the information and analyze it. So right now, biotechnologists and biologists, or molecular biologists, are collecting tons and tons and tons of information, and they can't analyze it all. So this bioinformaticist can develop computer programs that will do that and help these people understand their data. Okay, so lots of non-scientific positions. Okay, so we kind of already maybe talked about these. So sales, regulatory, so everything that's being sold, the FDA, which is the Food and Drug Administration. Okay, they're going to have a big role in this. Okay, they have to make sure that everything is safe and that there's enough of it available. Okay, you have to have lawyers, you have to have accountants, you have to have money, right? You have to have human resources, computer programmers, administration. There's security, a lot of security at these places. The more you understand about biology and biotechnology, the better these people are going to be able to do their job. Okay, so here's kind of how everything has changed over the past maybe, 60, 70 years. So if you look at this, I think this is just kind of amazing. So they call it snowballing because it just keeps getting bigger and bigger and bigger. Okay, so in 1940s, in World War II, the first antibiotic was produced, okay, and it was penicillin. So before the 1940s, there were no antibiotics. So if you had ear infection, strep throat, an infection with something that got cut, okay, there was no antibiotic to give you, okay. So 1940s, first antibiotics were produced. Okay, 1952, they figured out that, oh, look, DNA is genetic information. Everybody's got DNA. This is how information is going to be carried from one person to another or one organism to another. Okay, 1953, Watson Crick and Rosalind Franklin figured out the structure of DNA. Okay, in the 70s, we were able to take DNA, or not me, but they were able to take DNA and they were able to recombine it. Okay, so they were able to make new DNA, and then they were able to take that DNA and put it into E. coli. Okay, so we transformed it. We changed the E. coli. Okay, in the 80s, we had genetically engineered organisms start to be out there. Okay, so corn was a big one that was new. I saw my first genetically modified crops. It was in the early 90s, and they were in a little, the plot wasn't that big, okay, and it was all fenced in. It was like a prison, okay, so this is supposed to be my fence, and there was razor wire across the top of it, and they didn't want anybody going in there to mess with those plants. It was interesting, and now, right, they're everywhere. It's not a problem. Okay, so in the 1990s, we had started modifying animals. Okay, so if you look here, you have a cow. We have glowfish, right? You can buy the glowfish in Walmart. They're a genetically modified organism. Okay, now beyond, okay, so we've genetically modified all sorts of things. So we use it for agriculture, we use it for environmental research, we use it for medicine. We actually use it's industrial. So industrial, we're making enzymes that are going to go into detergents. So it's just kind of grown and grown and grown. So it's not going away. So the more that you can know about it, I think the more you're an informed citizen. Okay, so, whoops, this looks kind of weird. All right, so genetically modified organisms are called GMOs. Okay, and GMOs, it's just an organism whose genetic material's been altered. So how do we do this? Okay, and I love this because the press, okay, everybody makes it out to be a frankenfood, and then this is the picture I found. As soon as you put in a genetically modified organism, you get this stapled orange and apple put together, and it makes it look like Frankenstein, okay, frankenfood. Okay, but it doesn't look like that, okay? You probably don't even know that it's there. What you do is DNA is from two different places is cut, and it's going to be cut with this thing called a restriction enzyme, which we'll talk about more. And we recombine the fragments, we paste them together. Okay, we make this thing called recombinant DNA. Okay, and then what we can do is we can take that recombinant DNA and we can put it into a cell or an organism, and the organism will start to be able to do something that it couldn't do. Okay, so like the glowfish. The fish didn't glow under the black light before, but if we put this GFP gene in there, well, we haven't talked about this yet, but it's a green fluorescent protein gene, or gene that fluoresces. If we put that in there, okay, we can make the fish glow under a black light or a UV light. Okay, so sometimes GMOs are referred to as transgenic organisms. So a transgenic organism means we've taken a gene from one place and put it into another place. Okay, and that's kind of like the backbone of most biotechnology. Okay, so here's an example from the book. Okay, so we've taken a firefly. Isn't he cute? Okay, whoop. We've taken a firefly. Okay, and we found the gene that's going to make it glow. Okay, and it's luciferase. It's an enzyme. Okay, so luciferase is going to glow. So they cut it out of the firefly genome. Okay, and they could actually put it into a plant DNA. So they stuck it into the plant DNA. Okay, and then they took this and they stuck it into a plant. Okay, and they got a plant that glowed. Okay, and it's definitely not as straightforward as this. I mean, this makes it look like, oh yeah, piece of cake, we can do this. But it's really hard to get this to go from here, cut it out, put it into here, get it to work, okay, get it to be in the right order, and get it to glow. Okay, and then get it into a plant and get the plant to grow is not straightforward. It's not as easy as it looks. Okay, it's taken lots of researchers, lots and lots of time for that to happen. Plus, I don't know that this is necessarily a good example. Like, why do you want a glowing plant? Okay, so the idea is you could take this gene and you could link it up with another gene that you're interested in. And then if they're both there, you can kind of see the process. And I'll explain. This is just an introduction to get you started. Okay, so what kinds of products may we come across in our everyday lives? Okay, so all sorts of things. Okay, so I just want you to kind of maybe think about all sorts of different stuff. So there might be chemicals in your contact lens cleaner that's been produced using biotechnology. Corn is a big genetically modified crop. So like if you have tortilla chips or corn checks or cornbread, that's got a lot of them. Papaya, a lot of them are GMOs. Okay, so it's all over the place, and it's in all sorts of different things. Some of the enzymes that are in the laundry soap that remove stains are made using biotechnology. Okay, so a little bit about bioengineered products. Okay, so the first organism to produce human protein was, I said it before, it was E. coli, so Escherichia coli. So this is a bacteria. Okay, it's found everywhere. Okay, it's found in your gut. It's pretty common. Okay, there's a lot of different strains of it. One of the first things they did was they engineered it to make a protein, which is human growth hormone. Okay, so human growth hormone comes from your pituitary. Okay, another product that's out there is called TPA. So TPA is tissue plasminogen activator. Okay, so it's something that we make naturally, but we only make it a little tiny bit. Okay, so minute amounts. So we only make a little bit. What they figured out is what it does is it dissolves blood clots. Okay, so we have it normally, and what it does is it protects us from forming a blood clot where we don't want it. So they figured out, well, hey man, if we could make this in large amounts, then we might be able to help somebody that's having a heart attack or a stroke, or they have a clot where we don't want it to be. So it was one of the first biotech products to be sold. So you go to the hospital, they think you're having a heart attack or a stroke, or they have evidence of that. They'll give you aspirin. They might also give you TPA. So it's a clot buster. Okay, or some people say it thins your blood. Okay, so it's one of the first products to be produced using mammal cells. So it was produced using these Chinese hamster ovaries. Okay, so they took the ovaries out of these little fuzzy hamsters. They grew the cells from it, and then they put recombinant DNA in it, and got the cells to make a human protein. Okay, so like they don't kill hamsters over and over and over again. Okay, you'd buy the cells. So we'll talk about it. Okay, so here's an example of how they did it, the TPA. So if we go through it step by step, oh shoot. Okay, so they took a human, that's what it's supposed to be, and they got the TPA gene out of them. Okay, so the tissue plasminogen activator gene out. Okay, and then they had a vector, a piece of DNA that's just circular. Okay, and they cut this, and they cut this, okay, with the same, kind of to make the DNA look the same. Okay, with the same restriction enzyme. And then they could take this piece of the DNA, the green is supposed to be the human TPA gene, and they put it into the vector. Okay, and then they took the vector and they put it in the Chinese hamster cells. Okay, and they got the Chinese hamster cells to make the protein that we're interested in. Okay, so they call it recombinant RH, recombinant human tissue plasminogen activator, and that's clot buster. Okay, so I think that's the last slide. So this is kind of a wrap-up of, or a brief summary of the information in Chapter 1, and I'll post an assignment after this.